first example is the lock and key model, which basically just describes how the enzyme-substrate interaction works and showcases how an enzyme locks into a substrate at the active site using specific shapes, kind of like a puzzle. Anabolic reactions, like most enzyme reactions, follow the lock and key model. Anabolic reactions build a larger and more complex molecule through smaller ones. For an anabolic reaction, energy is required. Anabolic reactions are the reverse of catabolic reactions. A singular product is produced in this reaction. A catabolic reaction is when the substrates are separated into two separate products when they interact with the active site. During catabolic reactions, energy is released. The substrate is separated through degradation, and this is when polymers are broken down by bacteria. Feedback inhibition is when enzymes are suppressed by participating in a sequence of reactions. During these reactions, the substance is synthesized by the product of the sequence. The end product is used to block the activity of the original enzyme. During enzyme regulation, enzymes are regulated by other molecules. These molecules increase or decrease their activity. The four types of enzyme regulation are allosteric, genetic and covalent modification, and enzyme inhibition. Competitive inhibition describes when a substrate is blocked from binding to an active site by another molecule of a similar structure. Having a higher concentration of the substrate is beneficial because it gives the cell a better chance of a successful reaction. Non-competitive inhibition, on the other hand, describes when an inhibitor binds to the enzyme, which decreases the efficiency of the enzyme. The inhibitor binds to an allosteric site in the enzyme, which is separate from the active site where the substance binds. In the world of biology, every minuscule element serves a major component. Within the heart of a blooming flower, there is an extraordinary enzyme, sucrose. As we take a deeper dive into the stem of the flower and get to the cells, our exploration revolves around two main factors, sucrose and sucrase. Sucrose is a substance which is found in the flower cells and needs to be transformed into simpler sugars, a duo of glucose and fructose. These two sugars are later used as a source of energy to help with the growth of the flower and its metabolic process. This transformation only occurs with the help of sucrase, which is the key player of this process. It serves as a catalyst for a very specific biochemical reaction, which is critical for the flower's survival.
Now let's take a look at the heart of the sucrase's structure, the active site. The active site serves as the control center for the enzymes. This is where the magic happens. For example, the sucrase and sucrose come together to create simple sugars. This process is called the enzyme-substrate complex. The simple sugars later become the fundamental building blocks and provide energy. I hope you enjoyed learning about enzymes.